Okay, thank you. So yes, I would like to thank the organizer for having invited me to talk and my collaborators that are listed here. So I'm going to talk about the chiral puzzle of life. And first I will briefly then tell you what chirality is. So here is a brief reminder. Wow, I think it's okay. So you have here the basics, building blocks of life. So on the left, nucleic acids, and on the right, amino acids. And so life is based on carbon. So many molecules, they come with a central carbon, and the carbon is linked to 4 groups. And then that can come in 2 mirror images of one another. Actually, the word chiral is coming from Greek for hands. But when you do these molecules in laboratory, when you synthesize them, they always come in 50% of each left-handed and right-handed versions. This is because just they are chemically equivalent. However, from a biological point of view, they aren't. And life chooses to use only one chirality in these building blocks. So in nucleic acids, life is only using one type of sugar, so one type of mirror image and not the other. And then it's the same for amino acids. So the big question is how life made these types, because as I said, in laboratory experiments, and there were a famous one done in the 50s to understand how you synthesize the molecules of life in prebiotic conditions. And here they managed to get amino acids, but 50% of each versions. So I'm French, so I'm proud to show you the contribution of wine to scientific knowledge. So how chirality was actually discovered? It was by studying tartaric acid, sorry, I'm coming back, which is formed on corks by wine fermentations. And so Chiral was the first one to actually study this. Then he was followed by Jean-Baptiste Biot. And Biot actually put these crystals into solution, and he was witnessing that the plane of polarized light is turning. And he didn't know what was making the plane of polarized light to turn, but he said, it must have some molecular basis. So there were a lot of people trying to understand what was going on with what they call this optical activity. And then Pasteur made experiments on the same tartaric acid. And he had like one which was synthesized in laboratory that was not optically active. But he actually saw that there are 2 types of crystals in this racemic mixture. We call it racemic when it's not optically active. And this is its own drawing. So with a tweezer, he separated them. He put them into solution, and he showed to Jean-Baptiste Biot that one type of crystal was turning the plane of polarized light to the left and the other one to the right. And Biot was amazed really by this experiment. So, and also Pasteur witnessed that in natural substances, you only have one chirality. So he actually tried to explain it, and he made, I like to say, an anticipation, because that was written in 1848. And he said, if the molecules of life are asymmetric, it must be due to some asymmetry in the law of nature. So he postulated the existence of, sorry for this again. He postulated the existence of dissymmetric forces. And this was 1 century before the parity violation in the weak interaction was discovered. So in 1957, well, this is a well-known experiment of Miss Wu, but the point is that we have one force in physics that violates parity. So when you have, for example, a beta decay, then you don't have the equivalent mirror image in this world. This is because it involves a neutrino, which is a purely chiral particle. So when you have a decay, here it's a cobalt-60, then the electrons have a property because there are also neutrinos that are emitted, and they are spin polarized. So I'm a cosmic ray physicist, so I will show you the importance of spin in cosmic radiation. But just as a slide to show you that quantum mechanical effects should also be included in this chirality, is that the fact that the DNA and proteins use only one type of chirality in their building blocks this property allows them to adopt these stable helical structures. And DNA is the guarantor of genetic information, as you know. So you want this molecule to be stable, right? To be able to transmit the information that is inside it from one generation to the next. And this helical structure, it provokes quantum effects that are under study now and that are not understood yet. So 20 years ago, there is a phenomenon that was discovered by Ron Naaman at the Weizmann Institute of Science that actually helical molecules like double-stranded DNA or proteins, the alpha helix of proteins, they act as spin filters. And this could be very important for biological functions. So we don't know yet how to define life, but I wanted to show you that homochirality is really a signature of life, is unambiguous biosignature. And also, this chirality allows these stable helical structures that show some quantum effects that might be very important to understand how life is actually performing. So just these are like the facts that are linked to this chiral-induced spin selectivity effect. And we are trying to understand it, but again, there are no theory to really account for this observation. And so this is the optical activity that I was talking about. I don't want to be too technical, but it's related to the difference in absorption of left-handed polarized light to right-handed polarized light. So it's really chiral. And you see that both DNA and the alpha helix of protein show, interestingly, a peak around 192 nanometers. So this property of optical activity seems to be linked to the spin filtering effect, but we still have to understand this better. So anyway, so as I said, so I'm a cosmic ray physicist. I'm working on trying to understand the source of cosmic radiation and also to model how cosmic ray produces showers of particles in the atmosphere. So you may know that you are irradiated all the time by cosmic rays, but you may not know that actually there is one particle, the muon, which is dominated our radiation dose from cosmic ray. As I'm talking now, you are irradiated by muons. And these muons, they come from a weak decay. So the weak decay, the weak force is the one that is parity violating, and it's a two-body decay. So you have a muon which is coming from a positive pion or a muon which is coming from a negative pion. Both pions have a spin of 0, and they produce two-body decay of a neutrino, which, as I said, is a chiral particle, and a muon. And the muon, because of this is really a weak decay, will have a property. It will be spin polarized, 100% polarized in the rest frame of the pion, but on average at ground level, because you still have to do the Lorentz transformation, you have average spin polarization of the muon. So we are irradiating all the time by spin polarized cosmic radiation. And what we try to understand is actually if this spin polarized cosmic radiation could have had an effect that would have led to the emergence of a single chirality in the living organisms. So, of course, you could tell me, compared to natural radioactivity, right now, the radiation dose from cosmic ray may appear weak. But we have a supernova occurring in our neighborhood every like 5 million years. And so what we did is that we wanted to compare the radiation dose coming from spin polarized cosmic ray to the natural radioactivity, and this is what we actually did here. So so this is the radiation dose rate in milligray per year, and this is a function of time. So these are not my calculation, but this is showing you how the radiation dose due to natural radioactivity decayed over 4 billion years, and this is this is the natural radioactivity. This is the cosmic ray right now, the level of cosmic radiation right now. But this is what you would get from a supernova that would have occurred like 50 or 100 parsec from Earth. So this would really dominate our radiation dose by a few orders of magnitude. And again, the muons that would have been produced by this supernovae, just because the cosmic ray interact with the Earth atmosphere, they are spin polarized. So we try to understand better how actually you can, from the interaction of muons with biological molecule, they might have preferred one chirality over the other. So what we did is, as a disclaimer, I'm not a biologist, I'm a physicist, but we wanted to see if there is a difference in the damage made by polarized radiation if you have left-handed DNA and a right-handed DNA. So the electrostatic potential of a helical molecule is like a barber pole. And what we did is that we calculate the difference in ionization rate, so taking ionization as a proxy for mutation, between a left-handed and a right-handed helix. And we find a very small effect. But again, I mean, this is like a proof of concept. We have still to include the quantum effect that I was saying that may actually be important. But this is like inducing a very, very, very small difference in the mutation rate of one chirality, which would be more than the other. But then if you stick this into like evolutionary equations for the two populations, then you see that after a homochiralization timescale, which we would need to know what it is, right? I mean, it has to be short enough to be competitive, but then one chirality will dominate. So we were not the first, of course, to propose that maybe the fact that life uses one chirality is linked to the effect of polarized radiation. So I'm not going to go deeply into this slide because I don't have time, but it's just to show you a bit the state-of-the-art models on this subject. So basically, you're asking if homochirality is a prerequisite or a consequence of life. And by this, you're asking if you need already to have a prebiotic soup, which will be made on only the same chirality in their building blocks for life to be able to start. Or you say, okay, maybe life started anyway. There were like domains of different chirality and something that we don't know happened to make the transition from inert to living. But then the homochiralization is really a consequence of life because this biological function rely on spin. So then it's like it will be a preference toward one and not the other. So basically, this is what I'm showing here in this table. You have the prebiotic scenario, and many of them were actually investigated in laboratory. And so there is one which is actually showing that you have excess when you irradiate amino acids by ultraviolet circularly polarized light. You can get an excess of one chirality over the order of 3%. And so then you have to rely on some amplification mechanisms that could be at play. So you need this second step. So you can look at this publication. So you need basically a step which is prebiotic that will amplify everything to homochirality, and then you say, okay, from this homochiral soup, life managed to start. And then you have the biological scenario that I said, and the interesting thing is that it's actually never been investigated by laboratory experiments. So this is what we want to do. So we just started, so I have nothing really to report, but I can tell you the idea of what we want to do. We want to see if there is a different biological response of a living organism to spin polarized radiation. So what we can do is that we can do simple experiments to understand better how helical molecules interact with polarized radiation. So we can use UV-CPL spin polarized electrons or even muons. So we want to do it step by step. It's easier to start with light. And if we see that a different, for example, mutation rate is observed with left-handed and right-handed circular polarized light, that would be already something because these experiments have not been done. So in which environment you can find spin polarized radiation? So what we did is that we wanted to see in which environment this spin polarized muon would dominate. So muons are better than electrons because they retain their polarization until the moment they are captured by molecules, while electrons are very light. So even if they start spin polarized, they will scatter and lose their polarization quite fast. And here you see that these three components of the cosmic shower that I showed you before, so muonic is in solid line, electromagnetic is electron-positron, and this is the hadronic. You see that on Earth, at ground level, the muons are starting to dominate. On Mars, the atmosphere is very thin, so the shower has no really enough grammage to develop. And in very dense atmosphere like Venus or Titan, interestingly, in Venus, the muons dominate around 50, 60 kilometers above the surface. And this is like the habitable zone, right, for this planet. And the effect of muons, so I saw some talks last year that maybe some people think that there could be life in Venus clouds. So I said, even if it's not for homochirality, it's interesting to see what is the effect of muons on biological organisms, because here, for example, in this environment, this is the radiation dose from cosmic rays that will dominate. So, okay, should go faster. So we compared also Mars and the Earth. So again, I will go very fast, but Mars is a good environment because it shows signs that there were hot spring in the past, and hot spring is where we find actually the earliest sign of microbial form on Earth. So Dave Dimmer, sitting here, has a paper on this, and these hot springs can eventually make wet-dry cycles that will help to polymerize from primordial soup to actually make chains of longer and longer polymers. And interestingly, also in 2018, there were a lake, underwater water that was underground water that was reported. And if you can see the calculation here, again, on Mars, the muons start to dominate something like 10-meter water equivalent below the surface. So that means 10 meters of ice and like 3 meters of rock. And yeah, so I just wanted to point that. So we did it also for icy moon. It's not that different for Mars. So again, the muons will dominate underground. And I want to take I have 2 minutes on this slide, because so I think I managed to convince you that homochirality is a unique signature for life. So it would be very interesting to actually see if we can detect it, like detect chiral molecule. Maybe with these sun divers that can collect some data, or even amplifying, image of exoplanets. So what has been done so far on this is that we actually found some meteorites, class of meteorites, carbonaceous chondrites, that show an excess of left-handed molecule over right-handed molecules. So you can see this on this plot. So this is like even 20% level. So we never managed to get 20% in a lab. And but this is debated because you can still have Earth contamination. So what people wanted to do is to actually see in situ if we can find traces of excesses of one chirality over the other in organic compounds. So there was this mission, Rosetta, and actually Philae that bounced on the comet and found itself in the dark could not work, but one of the goals of the mission was to measure the chirality of the organic compounds. And now we have Hayabusa that actually came back 6 December 2020 with samples. The first report was that there is no chirality, no excesses of one chirality over the other. But they are still under analysis, and we have another mission coming from an asteroid, Bennu, in 2023, so Osiris-Rex, and also they will be able to analyze. But it would be great if we could find ways to actually detect chiral molecules. So, I don't have time to read this, but basically I said all this. So thank you very much.